Okay. I hope you have seen these topics for which I have uploaded for your first midterm. So you see that the all topics are from general embryology, and I want to go together with you today through this material and also discuss the images, diagrams, schematic images of the main stages of embryogenesis, which covers the weeks first up to 8 weeks. So we'll go to the look this one, third week of development. Last tutorial we have covered first 2 weeks. Now I want to go to the trilaminar germ disc. How do you remember how it is formed? Which type of main processes is involved in the formation of trilaminar embryo? Gastrulation, of course, gastrulation. But before, can you tell me, can you identify this image? What type of image is, for example, this one? What is this? Not what type, but what is this? Okay, zygote, definitely. And here we have two pronucleases. Yes, what has happened after this pronuclei fuse with each other? What has happened there? Okay, so first of all, it starts to divide. And this is two cell stage. This is two cell, these are two cells after the first division of the zygote. This is after the second one. And what type of structure is formed here? What is this? Morula. Okay. So the morula immediately after the third cleavage, the structure, the morula starts to undergo cleavage, compaction. Do you remember what compaction is? How it happens, what for compaction is, and what is the main results of compaction? So you see here, what type of process is represented here? How the zygote or already morula start to um, invade the uterine cavity, and what is here? What has happened here? Attachment of this type of structure, which is called blastocyst, start to invade the wall of uterus. So here we have already blastocyst. What is blastocyst and what are the main features of blastocyst? First of all, it contains cavity, and this cavity is blastocell. And how many populations of cells we have here? Two main populations which um, differ from each other. So one is here, inner cell mass, and another one covers the surface of the blastocyst. Yes, and this is trophoblast cell layer. What we have in the middle? In the middle, we have the cavity, and this cavity is called blastocell. Okay, so, and what we have here, or on the next slide, look. No, I do not need this one. I prefer this one here. Blastocell cavity, blastocyst itself, and we have here inner cell mass and trophoblast. So inner cell mass of embryoblast and trophoblast, blastocyst cavity. What has happened with this blastocyst at the end of this first week and beginning of second one? It start to invade wall of uterus. So this is uterine stroma, this is uterine epithelium. We have here trophoblast cells, we have here blastocyst cavity, we have here embryoblast itself. Please pay attention to the difference between the inner cell mass and trophoblast cells. Okay? What we have here on this slide. On this slide, we can see how the blastocysts start to invade more and more the uterine wall, and at the same time, these two populations of cells undergo differentiation, and they give rise to the another new populations of the cells. First of all, the trophoblast gives rise to the syncytial trophoblast and cytotrophoblast layers. Look, this dark green and light green layer of the cells. The inner cell mass of embryoblast, what has happened with it? It gives rise to two layers, yes? One is epiblast layer, stained blue, and hypoblast layer, stained yellow. We have here cavity, yes? How many cavities we have here? Cavity number one here, which is amniotic cavity, and cavity number two, which is blastocyst cavity. What is the first sign? So this blastocyst cavity then is transformed into the exocelomic cavity, which is or primitive yolk sac. So how it is formed, this exocelomic cavity or primitive yolk sac? What has happened here? Do you know what has happened here? So this exocelomic cavity or primitive yolk sac is formed, but how? So the cells of hypoblast proliferate, line this blastocyst inner surface. The blastocell became a little bit bigger, and finally it forms this primitive yolk sac. What you can see here, what else new in comparison with this stage of development you can identify here. You see that the what has happened with the trophoblast. So in the thickness of this layer, which is quite thick here, you can identify small spaces, and the small spaces are called lacunae. So these lacunae then fuse with the dilated and overloaded enlarged blood vessels of uh, maternal blood supply, and they fuse with them, and maternal blood start to move into the syncytial trophoblast. So where this, you see that syncytial trophoblast became thicker, yes? So which type of cells or which layer of this blastocyst provide formation of syncytial trophoblast? Of course, the cytotrophoblast, cells of which proliferate, move into the syncytial trophoblast, and here their borders, I mean borders of the cells, they are not more identifiable. So, okay, if you compare this stage of development with that one, what is the difference here? So the blood, maternal blood is moving to the blastocyst, plus new population of the cell is developed again, yes? So this new population of the cell gives rise to the, what layer is this? Extra embryonic mesoderm. And together with the formation of this extra embryonic mesoderm, it undergoes also vacuolizations. Look, these small spaces are derived in this layer, and only one small part is not vacuolized. So these all spaces fuse with each other, and they give rise to chorionic cavity. Yes, extra embryonic cavity. And what has happened with this, with the uh, bilaminar germ disc? It is still represented as a bilaminar, but changes happens in the yolk sac. So small parts of yolk sac pinched off, and yolk sac became much more smaller. So the whole this structure, embryoblast, together with this two cavities, is surrounded with a chorionic cavity or extra embryonic cavity. And the chorionic cavity itself became lined with a membrane, which is called extraembryonic somatic mesoderm. Look at this one, somatic mesoderm, which is chorionic plate. Another layer, this layer, covered the surface of embryoblast, I mean amniotic cavity and yolk sac, and gives rise to another leaflet of cells, I mean population of cells, which is called extra embryonic splanchnic mesoderm. Okay, This one. So this extraembryonic mesoderm gives rise to the chorionic cavity, one, and two leaflets of cell layers, or two layers of the cell. One is extraembryonic splanchnic mesoderm, and another one is extraembryonic somatic or chorionic plate mesoderm. Okay, this one and that one. So, what is this? This is this small part of extraembryonic mesoderm which does not under no, does not actually vacuolized, and it gives rise to the connecting stalk. So this connecting stalk is a later key player in formation of the umbilical cord. Okay, I think with these images here is everything understandable. If not, please write me. Should I repeat something or? Okay. So is everything understandable? We cannot see your screen. Okay, I can see it. Okay, I can see it now. Okay, so guys, maybe questions are with this um, stages, everything clear, hopefully, yes. If not, please write me. Okay, in this case, I think I can go further. So what is next step, next stage? How do you think? What should be the next stage of development? So by all these events which I have described now, the 2 weeks of embryogenesis, first 2 weeks are finished. And we have two-layered embryoblast, two cavities, two layers of extraembryonic mesoderm. So everything this gives us, mm, uh, due to everything this, we call this second week of development week of tooth, as you have remembered. So please, what happens with a trophoblast? With the trophoblast, okay, look please. Can you see here the trophoblast? Trophoblast is stained. Is stained. Look green initially. I'm talking about this layer. Yes. Look, layer of trophoblast cells. This is cross transverse section through the blastocyst. That is why you can see here cavity, inner cell mass, and trophoblast layer of the cell. So what has happened with these trophoblast layers? It starts to invade the wall, uterine epithelium, and at the same time, it undergoes further differentiation. So the trophoblast gives rise to the two layers. One is syncytial trophoblast, this dark stained one, and another one is cytotrophoblast. Okay, this one. So the cytotrophoblast layer, cells in this layer proliferate or divide and move into the syncytiotrophoblast. So this cytotrophoblast is the main source of syncytiotrophoblast layer. What is happened further with syncytiotrophoblast layer? What you can see here, lacunae are formed, these small spaces. Yes, and the small spaces later they fuse with the enlarged blood vessels or sinusoids in the uh, uterine wall, and by this way the maternal blood start to move. So the syncytiotrophoblast together with the cytotrophoblast and then extraembryonic mesoderm too, they provide formation of the um, blood supply, blood vessels. I will show you it a little bit later, okay? Is it understandable? So what has happened with the trophoblast? It gives rise to the syncytiotrophoblast and cytotrophoblast, and then syncytiotrophoblast gives rise to the lacunae via which the maternal blood start to move into these layers of the developing embryo. So is everything clear? In this case, I will go to another topic. So, third week of development and what has happened with the embryoblast during this third week. Okay. Look, please. This is a real image of embryoblast. So this is sample of embryoblast, and you can see here, look, this is the whole structure, bilayered embryoblast together with a primitive yolk sac forming extraembryonic mesoderm here. Look, small part of connecting stalk is visible here, and this is syncytiotrophoblast. Here, look, these spaces, lacunae, are clearly visible, and blood start to move in these small cavities. Plus, you can see here also layer of the cells, which is since cytotrophoblast. Look, this one. So hopefully you are able to identify here the cells. Do you remember I have told you that first what you have to do when you start to identify cells, you have to identify the nucleus, because nucleus usually using conventional histological stainings is always stained much more darker than the cytoplasm, especially when we are talking about the active cells. And you can see then, you can then much more easier identify the surrounding cytoplasm. So the surrounding cytoplasm might be clearly visible if it is, if the cell has quite big amount of cytoplasm and the ratio of nucleus to the cytoplasm is low. But if, for example, we have not only small rim of cytoplasm, in this case, identification of the whole cell, I mean borders of the cell, is not easy. So do you remember why I use this term borders, cell border, not cell membrane, for example? Look, Look here or here. I do not say that this is cell membrane, for example, cell plasma membrane. Or, for example, here, nucleus with a nucleolus and whole cell body is clearly visible. So this is because using the light microscopy, and this is light microscopic tissue sample. So you can't identify the cell plasma membrane. Do you remember why? Why it is impossible to see in light microscopic image the cell plasma membrane? Please write me this, okay? Why? So why the cell plasma membrane is not identifiable using the light microscopy? I mean conventional light microscopy, optical microscopy. What is here the most important parameter which plays the key role? Have you heard about the resolution? Do you know what resolution is? Resolution power of the microscope. Do you know? Yes, of course. Little, too small resolution power. And when we can, for example, using which type of microscopical technique we are able to identify cell plasma membrane? Can you write me? Yes, exactly. Electron microscopy. So, but we can see this borders of the cell. Yes, we can see the cell body even in the light microscopy. So how it happens? We can see the cell membrane and attached macromolecules from outside. That is why the borders of the cell are clearly identifiable here. Okay, tell me please, okay, let's go back to our presentation. Yes, here. Okay, with this slide, I think everything is clear. Yes, fully implanted human blastocyst, and here are possible to identify this maternal blood cells in the lacunae. And you know these images, yes? So another sample of um, embryoblast with a yolk sac, and this is already formed chorionic cavity. Look how small is yolk sac in comparison with the chorionic cavity. And plus, you can see here the syncytiotrophoblast, cytotrophoblast, which start to form primary villi. So what primary villi are? Look, primary villi are these protrusions of the trophoblast. Look, this one here, cytotrophoblast. And if it contains the extraembryonic mesoderm, somatic mesoderm, in this case, it is secondary villus. If not, if it contains only syncytiotrophoblast and cytotrophoblast, this is primary villus. And later, this mesodermal core start to form the blood vessels, blood capillaries. And this structure is, after the formation of these blood capillaries there, blood vessels, became tertiary villus. Okay, so the villus capillary system is finally formed by the end of the third week. But before this, together with formation of this different type of villi, the very important processes are developed in the embryoblast itself. So gastrulation, as you remember. And what is the first sign of gastrulation process? What actually the gastrulation is? Gastrulation is the process that um, via which the all three germ layers are formed: ectoderm, mesoderm, and endoderm in the embryo. And what is the first sign of gastrulation, that the embryo undergoes gastrulation process? Formation of primitive streak. So the primitive streak is a narrow groove, and which on the both sides has slightly bulging mm, folds of the epiblast. So I'm talking about this layer. And the primitive streak is developed on this surface here, which is the bottom of the amniotic cavity at the same time. And besides the um, indication that the gastrulation process is started, what is the importance of primitive streak? Why formation of primitive streak is very important? What we can identify after the primitive streak is formed? We can identify poles of the embryo blast. Yes. So the primitive streak is usually formed in caudal end, and simultaneously with the primitive streak, oropharyngeal membrane is also formed. So the oropharyngeal membrane is the um, place where the epiblast and hypoblast are tightly attached to each other, and they give rise to the membrane like structure here. So in the so primitive streak is formed in the caudal end. Oropharyngeal membrane is formed in the cranial end. Primitive streak itself has two ends itself. I'm talking about the primitive streak itself. So the um, small, the end, cranial end of primitive streak is this one. And here the primitive node is formed too. So the primitive node consists of a slightly bulging area and pit in the middle, small indentation, and at the most caudal part, a little bit later, cloacal membrane will form. Okay, so this is surface of what? Surface of epiblast layer. This is epiblast layer, and this is surface of epiblast layer. And this one is yellow. This yellow is hypoblast layer. So this part is removed from bilaminar structure from this embryoblast, and you can't see here yolk sac. Yolk sac should be here, and this is the wall of yolk sac, and this is wall of amniotic cavity. Small parts, of course, of their walls. Is everything clear with this? Is everything clear? Okay, in this case, I go further. So what is depicted on this slide? What is described on this, depicted on these two images? I mean, the upper one and the lower one. Gastrulation process itself. So what gastrulation means, as you know, formation of this trilaminar layer, trilaminar embryo, and the first layer which is formed during the gastrulation process is endoderm. Yes? How it is formed? Endoderm. What is from which layer of bilaminar germ disc or bilaminar embryoblast it forms? From epiblast. And what has happened with the epiblast cells? Initially, they are tightly attached to each other, yes, but later they are detached from each other and start to migrate, move down beneath the primitive streak. And due to the movement, these epiblast cells, former epiblast cells, start to occupy this middle part of hypoblast, push these hypoblast cells to the periphery and occupy finally the whole epiblast layer and give hypoblast layer and give rise to the endoderm. So the a definitive endoderm is formed first as a result of first wave of cell migration. So the second wave gives rise to another layer which occupies the space between the epiblast and already formed endoderm. And it gives rise to the mesoderm. And then later the rest of cells, which is representing in the epiblast layer, they became ectodermal cells. And here, the movement, control of cell movement is uh, regulated by, movement is controlled by the E-cadherin molecules, which undergoes down regulation. It means that they are, you know what down regulation means, that the expression became lower. And this is the protein which normally attach these epiblast cells to each other. Okay, another event what is developed in this trilaminar during the formation of trilaminar embryo are formation of different types of structures which serve as a reference points of embryonic disc. So the first one is oropharyngeal membrane. And I have mentioned already that oropharyngeal membrane is formed always at the cranial end of the embryo blast. and this actually is result of tight attachment of ectoderm to the endoderm. Plus, we have here also, in the future, here will develop the oral cavity. Another one is cloacal membrane, which is formed at the caudal end of primitive streak here. And again, here we have tightly attached ectoderm to the endoderm layers. Another one is precaudal plate. So the precaudal plate, it is, look, you see here these arrows. What these arrows are? What these arrows indicate? They indicate a movement, route of cell migration, direction of the cell movement. All of them, the most part of cells moves cranially. Look, here, here, here. Only small population moves from one side to another side uh, laterally. And you know this movement of the cells, first their location and then direction of their movement determines fate of these cells. Look, this is a map actually, which represents the primitive streak population of cells, different population of the cells in their fate map. Fate map of epiblast cells. We have here cloacal membrane, we have here oropharyngeal membrane, and here these different populations of the cells are indicated. So this population of the cell, the most rostral in the primitive, which surrounds the primitive node and moves rostrally, they give rise to the notochord. Look, this layer. So notochord is layer of the cells which is developed in the mesoderm by the migration of epiblast cells between the epiblast layer and endoderm layer. And finally, when the mesoderm is formed, they give rise to the notochord. Notochord. Look, I'm talking about this structure, notochord. So, uh, look, this is dorsal surface view of embryo, and the surface of epiblast is clearly visible here, and we can identify, and the different populations of the cells are depicted here. So the most cranially allocated population of the cell, which start move forward into the cranial direction, it gives rise to the notochord, to this structure. And but before notochord is formed, precaudal mesoderm is formed, and the precaudal mesoderm surrounds the cloacal, oh sorry, oropharyngeal membrane. So look please here, oropharyngeal membrane. So very tight attachment of two layers, ectoderm and mesoderm. This is oropharyngeal membrane. The same is here, again, tight attachment of ectoderm and endoderm, you have to pay attention to the colors, because later when the different type of germ uh, organs, I mean germinative organs, start to form, they are indicated with the appropriate colors. I mean colors of layers which gives rise to them. Okay, so cloacal plate, which contains, or cloacal membrane, which contains this um, layer of ectoderm and endoderm, and the small protrusion of which layer, endoderm layer, gives rise to the allantois. So the small protrusion is identifiable in connected stalk. Okay. So allantois then will participate in formation, this further differentiation of connecting stalk, and then it undergoes, of course, degradation. But this is surface view, yes, of uh, epiblast, which then is differentiated into the ectoderm. And you see here all these movement directions. And first one about which I start to talk with you was um, precaudal plate. So this precaudal plate forms around the oropharyngeal membrane via the movement of cells in cranial direction, but at the same time from the cranial portion of the primitive strip, which is called primitive node or the organizer, the small population of the cells start to move forward in cranial direction. They initially give rise to this prenotochordal cell population, which then moves further and differentiate and gives rise to the notochord. This is surface view and this is sagittal view. Do you remember what is the sagittal view? Sagittal view or sagittal section is section which is parallel to the midline section. Okay, midline section. Is everything clear with formation of uh, precaudal plate and notochord? Is it? Okay, excellent. So, um, the next was, I go back to the map, fate map, and so we have already discussed the small population of the cells, which is derived from primitive node and which gives rise to notochord. Notochord is very important structure. This is the main structure which induces the formation of uh, neural plate later, neural tube, and finally, this neural tube gives rise to the nervous system, to the central part of nervous system, I mean brain and spinal cord. Another population of the cells, which is indicated here with all these layers, look, this blue, light blue, purple, and green, they give rise to the mesodermal structures. Look, all of them. Only this yellow stained population of the cells gives rise to the extra embryonic mesoderm. Okay, this one. Do you remember extra embryonic mesoderm, which gives rise to chorionic cavity and two layers, two extra embryonic mesodermal layers. One is somatic layer, which covers the inner surface of chorionic cavity and gives rise to the chorionic plate. And another one is splanchnic layer, which covers the surface of the yolk sac and amniotic cavity and gives, and this is splanchnic extraembryonic mesoderm. Okay? So all other these layers look indicated with the PM, IM, and LPM, they give rise to the mesoderm. And mesoderm itself, look over here, mesoderm, no, sorry. It should be in another, I mean, further differentiation of mesoderm. So this is what type of section? Transverse section. Yes. The previous one, when we see, for example, the length of notochord, and we see the precaudal plate, and we see the, for example, the um, whole length of ectoderm and mesoderm and endoderm. So it was the sagittal section. This is transverse section. And in this transverse section here, you can see how the um, mesoderm is differentiated. So this is mesoderm, this red stain, and this mesodermal layer is quite thick. Here, at this stage, we have here endoderm, which is covered with a layer of um, mesoderm, and initially it is extraembryonic mesoderm, and here is extraembryonic splanchnic mesoderm too. But later, when this intraembryonic mesoderm is start to differentiate, it gives rise to the three main parts of mesoderm. So this is paraxial mesoderm. Look, this one, paraxial on the both side of um, main axis of the body, developing embryo. And then here start to develop small part, intermediate mesoderm, and then lateral mesoderm. So initially this lateral mesoderm is whole structure and is in continuum with the splanchnic extraembryonic mesoderm, but later in this lateral part of mesoderm start to develop cavity. Look, small spaces, and the small spaces fuse which is other and give rise to the little bit more, I mean, the volume of cavities. And finally, this lateral mesoderm is divided in two layers. So the one layer became visceral mesoderm layer, this one, or splanchnic, and another one became parietal mesoderm layer. Look, this one, which covers the surface of amniotic cavity and the amnion. So trilaminar embryo undergoes the further differentiation. And immediately, what is what makes embryology a little bit um, complicated subject, and I mean for study, um, that all these processes which are described um, separately, they actually are developed simultaneously or overlapped their frame of time during which they are developed, they overlap each other. So formation of ectoderm, formation of notochord in the mesoderm. So it stimulate formation of neural plate in ectoderm. And immediately when this some part of the ectodermal cells became neural plate cells, this layer gives rise to the folds. Look to these two folds. And these two folds rise up step by step, and finally they give rise to the groove on the surface of the ectoderm, which is called neural groove. And these folds finally fuse each other. And after their fusion, they give rise to the neural tube, which is separated from the ectoderm, and the mesoderm further differentiate into the somites. So the paraxial mesoderm gives rise to the somites, to the intermediate mesoderm, and this small cavity, which initially was start to develop on the 19 days of at the end of third week of embryogenesis, this cavity is actually the primitive intraembryonic cavity, which then became bigger. Here, look, this one, intraembryonic cavity, body cavity. And finally, this, via the process of folding and fusion of these two folds with a chorionic, with a yolk sac and with each other, it depends on the part of the body, they give rise to the body cavities. Okay? Is here everything understandable? I mean, the formation of, for example, neural tube and um, intraembryonic cavity. Is it understandable? If not, I can repeat, no problem. So tutorial is for explanations, for questions, please. Is everything understandable? Okay, excellent. Um, Maybe you need break. I can give you 5 minutes, not more. Okay? 5 minutes. You can take a break. Fine.
Can you check the question, please?
So, can we start?
Can you check a question?
Question? Which question? Okay, I will see. Okay. So I have, please, the midterm exam for body 1, including histology and anatomy, will be the same exam paper in the same, yes, the same exam, the same day, as I know. Is lecture 1 the microscope and lecture 2 the gut tube and body cavities of the midterm? Um, you know, yes, actually, the gut tube and body cavities will be included in the, or we can include it here too, gut tube and body cavities. So your midterm is when? It is May 8th. Yes. So, yes, it will be without gut tube and body cavities. Only these topics of general embryology, which I have listed there. Okay. So, lecture one. You can see these topics. I can show you, of course, but I have uploaded it on the drive, and you can see this on the drive too. So, the first week of development with the main results of fertilization and cleavage and implantation and blastocyst formation. Then the second week, formation of bilaminar germ disc. Third week, of course, trilaminar germ disc formation. All pages are indicated here, what you need to read. Then the process of gastrulation, fate map, okay? And then the further development of the trophoblast, 38 weeks, the embryonic period, when the ectodermal, mesodermal, and endodermal layers start to form the um, germinative organs. Okay, almost all organ systems became formed, of course, at the germinative level. So this is the main topics which you have to study for midterm one. Okay. Together with this images, I mean diagrammatic images of these different stages, I upload this set of images on your drive too, on Google Drive, so you will see that this. Or, you do not need even this set of images. Please simply pay attention to all these images, because these images helps you to understand the processes, and at the same time it will facilitate your thinking and your answering. I mean, answering of your questions during the midterm. So, for example, it might be here the image where some of these terms will be removed, and they might be enumerated. And for example, the question might be uh, what is indicated with a number 1 here, or what is indicated with the number 2 or 3 or 4. Okay, so if you have studied the material, if you have read it, read it, so in this case, everything this will be easy identifiable. Okay, let's go back to our topics, to our fate map. Okay, so what was here? So this hopefully you understand that here is talk about the ectodermal cells, or first epiblast layer cells, which proliferate and then start to migrate. And they migrate in cranial direction. They migrate beneath the primitive streak. They migrate even on the other side of the embryo. So by this movement, these epiblast cells, they form mesodermal layer. The first wave, the initial wave of migration, which gives rise to the extraembryonic mesoderm or chorion, chorionic cavity and chorion plate later, is started here. But by this wave of cell migration, the extraembryonic mesoderm is formed. Extraembryonic mesoderm. But later, when the cells start to move, start to migrate, they form the endoderm, correct? Then mesoderm, and then the rest of the cells became ectoderm. So this the most cranial portion of the cells from primitive node moves forward and gives rise to the notochord. Here we have precaudal plate. Do you remember? And this precaudal plate and notochord, they provide formation of uh, first of all notochord, of course, of neural tube, but formation of uh, the brain, the most cranial part of this embryo, is of course determined by the precaudal plate formation, which surrounds this oropharyngeal membrane, especially forebrain. Formation of forebrain is affected by the um, precaudal plate. So this process, when, for example, some set of cells via the signal molecules are able to change the fate of cells or determine the fate of cells, is called induction. So this typical induction process takes place in the ectoderm when the via the induction influence of notochord, the neural plate is formed in the ectodermal layer. So the paraxial mesoderm, do you remember paraxial mesoderm on the both sides of embryo, both sides of neural tube, which gives rise to the somites? Yes. And then is the intermediate mesoderm, and from intermediate, this paraxial mesoderm forms actually the vertebra somites and somitomeres, which then participate in the formation of the um, skeletal system and muscles too. But the intermediate mesoderm, do you remember intermediate mesoderm? So intermediate one, it gives rise to the urogenital system. And here in the lateral mesoderm, these small cavities are developed, and small cavities give rise to the body cavity. And this parietal and the visceral layer of the mesoderm, look this one, they form the organs, that is visceral organs of the human body. So this is forming neural tube, neural groove, neural folds, finally formed neural tube, and here we have paraxial mesoderm, which gives rise to the somites. So this is transverse section through the embryo. Yes, but another, for example, this one. What type of section is this? This is sagittal, parallel to the midline section. Okay, so somites looks like these structures here. Look, somites, and this is neural tube. Usually it is covered with the ectoderm. Look, this is ectodermal layer, and the ectodermal layer is here removed to demonstrate the surface of neural tube and formation process of somites. Look, not all somites are finally formed. They are in the formation process. So here, these one are clearly identifiable, but here in this part of mesoderm, it undergoes still formation process. And these somites, which are formed from paraxial mesoderm, they are clearly visible on the surface of embryo here. And this diagrammatic image, look, this is neural folds, this one and that one. I'm talking about these folds, this one and that one. And then they rise up, fuse each, and start to fuse each other. In the cranial part, these folds are much more wide, and in the trunk part, they form somites, and at the same time, fusion of these folds is initiated here. And as a result of fusion, the neural tube is formed, and the somites, I'm talking about these structures, okay? Look, somites, they start to differentiate. And via the differentiation, they give rise to the, finally, they give rise to the very important structure, myodermatome and sclerotome. And this dermatome gives rise to the skin of a back. The sclerotome forms, look, the sclerotomal cells, they form the vertebra, ribs, and the myotome participate in the formation of the muscles. At the same time, this small portion of the cells, look, which is allocated on the in the dermatome, in the ventrolateral part. This is a dorsomedial portion. This participate in the formation of sclerotomal cells and myoblasts also. But the small population of the cells, it moves into the parietal layer of mesoderm. This is intraembryonic cavity. Do you remember this place in the lateral mesoderm? So in the lateral mesoderm, when I'm talking about this portion, this part, okay, or this cavity, this part. So this small population of the cells moves into the parietal layer and later will participate in the formation of lymph buds. So besides the visceral mesoderm layer, look, visceral mesoderm layer. This was parietal, okay? Parietal, which was in continuum with the extraembryonic mesoderm. And here is visceral one. So the visceral mesoderm together with the endoderm, endoderm, it forms the wall of gut. And in the rostral and caudal part of the embryo, these layers fuse each other and give rise to the gut tube. But in the middle part, where the uh, middle gut is connected with the yolk sac, it became vitally duct. This is due to the folding process. Folding. So the embryo undergoes folding. We have here two processes: lateral folding. This is lateral folding, and another one is so left and right. Cranial part and caudal part is not determined, not depicted on these images. So the body cavity, intraembryonic body cavity is formed, gut tube is formed, and gut tube contains this visceral mesoderm layer and also endoderm layer. And you also can see here mesentery, dorsal mesentery, which is actually continuum of extra of visceral and splanchnic mesoderm, and this dorsal mesentery provide attachment of gut tube to the wall of body cavity. Look here. So what we have here amniotic cavity. Look, these two folds go down, fuse each other in the rostral and caudal part of the embryo, but not in the middle part. In the middle part, they fuse where the midgut is. They fuse with the yolk sac, and they form the cavity, which is the intraembryonic cavity. Okay. So, plus the mesoderm is able, mesoderm has the capacity to form the blood vessels. And the blood vessels are formed by two main ways. So, first one is vasculogenesis, and during the vasculogenesis, the mesopopulation of mesoderm cells differentiate and gives rise to the hemangioblasts. And these hemangioblasts, they finally form the blood cells and the blood vessels. Look, both of them, or uh, it might be due to the vasculogenesis might be due to the angiogenesis. So it means that the pre-existing blood capillaries, they give rise to the new one. And this everything is formed, of course, in the um, extraembryonic mesoderm, in the villi, and that is why the villi undergo differentiation, and finally they form a tertiary villus, which is in uh, direct connections with the maternal sinusoids, and wall of yolk sac. Also, in the wall of yolk sac, the blood vessels are differentiated. So this is small pericardial cavity here. Here is oropharyngeal membrane, do you remember? And here the mouth cavity will be formed, pericardial cavity. And when these two walls come closer and closer to each other, they finally form here the gut tube. Okay, so these are the main transformations, main differentiation routes of mesoderm. What has happened with the mesoderm during the from the third up to the eighth week. So, what has happened with the ectoderm or endoderm? So, with the mesoderm, is everything clear? Hopefully, and do you remember this fate map? I think the fate map is also understandable in this case. You know what the paraxial mesoderm is? Look, this both parts, the most wide part of mesoderm on both sides of neural tube, which gives rise to what? To somites, and then is intermediate mesoderm, which is formed from this population of um, epiblast cells, and this population of epiblast cells gives rise to the intermediate mesoderm. What has happened with this population of cells? They migrate, they detach from each other, and they migrate down beneath the primitive streak. The same has happened with this paraxial mesoderm. Only via the movement beneath the primitive streak, paraxial mesoderm will be formed. And then this is lateral plate mesoderm or lateral mesoderm. And this also moves down here beneath the primitive streak and forms the most lateral part of the mesoderm. So what has happened a little bit later with this lateral plate mesoderm? The small cavities are formed there, they fuse each other, and what is formed the initial steps of intraembryonic or body cavity. And this step by step, due to the folding, then this body cavity is closed, finally formed. This we will see a little bit later. So hopefully with this arrows is also everything clear. Yes, so they depict how the direct movement directions of the cell populations. So this is mid-sagittal sagittal section. This is transverse section. That is why you see here only ectoderm, mesoderm, and notochordal plate. So, and here intraembryonic mesoderm, of course, not extraembryonic, intraembryonic mesoderm, and part of this, depending on in which part of the embryo this section is done, it might be precaudal if the section is done here. If section will be done here, we will see what? We will see here the notochordal plate and intraembryonic mesoderm. If it will be sectioned here, we will see here only the mesoderm layer. Okay? But later, when the neural tube will be formed, we will see the end part of neural tube too. Okay, so you see here at the end of fourth week how the a trophoblast is differentiated into the uh, tertiary villi, and finally, in tertiary villi, the extraembryonic mesoderm gives rise to the blood vessels, and these blood vessels finally uh, are um, continuous with the blood vessels formed in the embryo itself. So this will be, of course, the further uh, umbilical, future umbilical cord, and this will differentiate it into the placental derivatives. Here is chorionic cavity, of course. Look, this is chorionic cavity. This is yolk sac, definitive yolk sac, and this one is amniotic cavity. And here we have this uh, trilaminar embryo. At this step, we have here bilaminar. A little bit at the end of third and beginning of fourth week, we have definitely trilaminar embryo here. Okay, so what is happened with the ectoderm further? So the neurulation process is initiated. So what is the neurulation process? Look, we have here this surface image of ectoderm. Is everything clear with the mesoderm? Is it? If not, please feel free, ask the questions. Yes, the exam will be multiple choice. Spend time looking at these slides. Which you mean? Which slides you mean? So what is the topic? You are talking about the presentation. So this presentation is prepared from your textbook. I mean, these images are used from your textbook. If we carefully read the textbook, these indicated pages, of course, to understand everything what is written there, you have to pay attention to the images. And of course, you will know this, how to interpret these images if they will be included in your questions. Okay, so we do not need, the exam will be, yes, multiple choice. Lecture 1 and lecture 2. So the lecture 2 is body cavity. So it is enumerated in your portal. So the lecture number 1, okay, with microscope, of course, techniques of microscopes, but the lecture number 2 should be the initial steps of embryogenesis, formation of bilaminar discs and trilaminar discs. The formation of gut tube and body cavities was the last lecture in the general embryology. Okay, I go back. Is everything clear? Can I go to the presentation back? Okay, in this case, so neurulation. Uh, we have here surface image of epiblast layer. Yes, formation of primitive streak, formation of primitive node again. Here, a little bit later, formation of local membrane. Here we have oropharyngeal membrane, but on the surface of ectoderm, epiblast, or already it is differentiated into the ectoderm, the population of part of cells undergoes differentiation due to the induction of the notochord, from the notochordal cells, and it gives rise to the neural plate. Look, these are ectodermal cells which became able to express appropriate genes for formation of neural tube. That is why these folds on both sides, they rise up step by step and form this neural groove. Look, this is a real embryo here, view of real embryo. What you see here is primitive node. Look, this image actually is represented here. So, I mean, the schematic image, a schematic image of this part of embryo. You see here the primitive node, primitive streak, cloacal membrane, cranial end of the embryo. And the whole this part is yolk sac. Okay? Yolk sac. Here you see the neural plate. Look, neural plate. And look the small parts. So this is process of somite formation, initiation of somite formation. And here the somites became clearly visible. But neural groove is still represented here. And then they fuse with each other, I mean folds, and somites, formation of somites goes in cranial and caudal direction, farther, of course. And step by step, this cranial part and caudal part is fused with each other. And look, this schematic image depicts, actually is the diagrammatic image of this stage embryo, 22 days. It means end of third week. The same is here. I mean, diagrammatic image of this one. And here we have neuropores, anterior neuropore and posterior neuropore. You know, this is place where the neural tube is opened and the amniotic cavity moves into the amniotic fluid moves into the neural tube. Okay, so you see here the somites formed and somitomeres start to form. So these segments of mesoderm gives rise to the segments in the same style. The neural tube undergoes segmentation, and this neuromeres, it forms the neuromeres. So in anterior part, the somitomeres together with the neuromeres start to form the brain. And from this part of the neural tube, of course, the spinal cord is formed. Then these pores are closed approximately at the end of fourth week. And initially at the 25th day, what we have here anterior neuropore. Do you remember? I'm talking about this one. Then posterior, again, I am talking about this one. So at the end of fourth week, they become closed. And what we have here, we have pericardial bulk, where the heart finally will be differentiated and formed. We have here also the connecting vital duct, which forms a connection between the uh, gut tube to the yolk sac. So you see that body cavity is closed in the anterior part of the embryo and posterior part. But the middle part, where the midgut is formed, it is connected with the yolk sac. So via the folding, these two folds fuse each other in the anterior part and posterior part, but they fuse with the yolk sac in the middle part of the embryo. Okay, is here everything understandable? And look, due to the craniocaudal folding, so beside this lateral folding, this cranial and caudal portion of the embryo also folds. It means it moves down, and this is via the due to the proliferation of the cells and formation and rising amount of cells in the developing embryo. So you see here this look these rod-like structures. These are pharyngeal arches, and pharyngeal arches are very important type of temporary structures. They are differentiated from the mesoderm. So when you look at the embryo, what layer covers the surface of the embryo? Of course, ectoderm. Beneath is mesoderm, and this mesodermal tissue gives rise to the um, pharyngeal arches. But here I have to mention another germ layer or of the population of the cells, which is derived from the ectoderm. Hopefully you know what type of process is depicted here. Trilaminar embryo, ectoderm, mesoderm, paraxial mesoderm, intermediate mesoderm, lateral mesoderm, parietal layer, and the splanchnic layer, intraembryonic cavity, yes, notochord. And here you see that the ectoderm undergoes folding. So it gives rise, it differentiates into the neural plate. And this neural plate then gives rise to the folds, folds rise up, they fuse finally each other, but before they fuse, these ridges of the folds gives rise to the absolutely new population of the cells, which is called neural crest cells. And this portion of the fold is called neural crest. So this population of the cells, when these folds fuse each other, start to migrate. Look, these arrows indicate the uh, migration route of these neural crest cells. And after the migration, these cells participate in the formation of pharyngeal arches. When they move into the rod-like thickenings of mesoderm, finally the mass of these rods rise up, and between these rods the indentations are formed. And via these indentations, this first, second, third, and fourth, fifth, and sixth one are identifiable, but very soon this fifth, and sixth ones, fourth, fifth, and sixth ones, they fuse each other. At the same time, they give rise to the dorsal root ganglia. This is part of peripheral nervous system. They give rise to the melanocytes, pigment cells in the skin. I'm talking about the um, neural crest cells again. And they give rise to the adrenal medulla, they give rise to the sympathetic ganglia. So they are very quite big. They are population of the cells with quite big competencies. So, and before because they differentiate into the many different type of structure, they are called as well, as a fourth. They are mentioned as a fourth germinative layer. The first three one are ectoderm, mesoderm, endoderm, and this is the fourth one. You see how they these neural crest cells give rise to the ganglia, to the gland part, and to the enteric ganglia. I mean, nervous innervations, ganglia in the gut tube. You know, the intestines have very, very strong innervation. Huge amount of neurons are represented there. So many, almost the same amount of neurons as in the brain we have is represented in neural in the GI tract. So that is why it is called the second brain. But you know what is interesting? This maybe due to this high innervation, very interesting is influence of GI tract on the proper functioning of the brain. Okay, guys, I'm done with the ectoderm. I'm done with the endoderm, with the mesoderm, derivatives of all these three structures. Hopefully, everything is understandable for you. If not, please write me the questions. I will, of course, re-explain and clarify everything what you want to know. So, any questions? Everything is clear. What is clear or not? Yes or no? Okay, yes. In this case, please read the topics which are uploaded on the drive for your midterm. Read carefully the indicated pages. You do not need for the midterm the pathological processes which are described, no molecular regulations because you have not enough knowledge of molecular biology and biochemistry. This everything will be you will study in the future together with the medical genetics, etc. So but please pay attention to these main processes and pay attention to the diagrammatic images, because these main diagrammatic images will be included, might be included in your questions, okay, in your questionnaire. In this case, if everything is clear, please once again confirm that everything is clear, okay? Yes or no? In this case, I say you bye and meet you next week. So after 7 minutes or 8 minutes, you have the anatomy. Bye, guys. Will there be any histology in the meter? No, only embryology. We have not started histology yet. Bye, 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 bye. Thank you for your attention.